Thanks, Don and Liang. So Dr. Zhao will ask you to pivot for workshop summation remarks. As everyone knows, Dr. Zhao is Director of the Division of Quality Methods and Modeling in the Office of Research and Standards in OGD. Thanks for providing workshop summation, Liang.
Thank you, Jim. Actually, my summary is mainly to honor and show our appreciations to all the outstanding speakers who have critically contributed to the success of the workshop today. Yesterday, Dr. Lucy Fang did a summary. Today, out of respect, I want to do a quick summary on the learnings from this morning and afternoon sessions. Basically, we have two parts from today's talk. The first part is based on the oral PBPK modeling, impact, and needs, and opportunities, challenges. The second part is model master file, which we just closed the session. And I appreciate everyone still staying with us on late Friday. And I will try to get my summary as short, as succinct as possible. For the drug absorption, it represents one of the most fundamental sciences for pharmaceutical scientists and the industry. The majority of the drug products are orally administered, and any breakthrough in science and modeling will have a long-lasting and high impact to the field. I want to borrow some kind of a thought from Zhao presented in the morning regarding the need from the oral PBPK modeling. Basically, they are all bearing high regulatory impact in terms of meeting industry's need, shortcut, abbreviate the regulatory program, and also meet some of the ICH harmonization requirement. The need includes the bio-waiver for post-approval changes beyond SUPAC level 2 requirement, the bio-waiver for non-proportional formulation, and the waiver for Fat B studies, the bio-waiver for BCF class 3 drugs for non-Q1, Q2 compliant formulations. We also have, they are perfectly aligned with our internal kind of thinking on the ICH need as well. We have already launched some internal research in all of these areas. The challenges I heard from this morning is the trust on PBPK modeling outcomes. The physicians or other, even including some regulatory bodies, they think the model is too complex. Simulation may be performer dependent. Therefore, there could be a confidence issue on the modeling outcomes. The other challenge could be the software performance issue. The model validation can be very demanding. Sometimes the data needed for model verification and validation can surpass the need just to conduct the actual study. The model, sometimes not verified sufficiently for different formulations, and certainly the variability, for example, the within subject variability have not been fully characterized by PBPK modeling. There is still research gap in that area. I think along with the modeling verification and validation, then it's also logistically difficult to develop another formulation to verify, validate a model. Sometimes we know it's costly to develop a non-BE batch. Also, we learned that for food effect, we have moderate or low confidence to use PBPK models, especially for BCS2 and it didn't mention this, but by detection, I think it's also difficult for BCS4 compounds. Whenever we have a good understanding on the driving mechanism of the food effect, we may gain high confidence. For example, if the formulation is only impacting on the effect on the luminal fluid or the physiology of GI tract, then we may have high confidence on the model simulated outcomes. Then we also learned that, from the IQ investigation of 30 compounds, and it already generated several key publications, which lent us good knowledge and experience in terms of understanding food effect. However, it has not included the formulation as a factor for these 30 compounds. And certainly more input of consideration on the mechanism of food on this compound on the formulation in terms of PK exposure will certainly gain us more confidence. And we also learned that the propagation of within substrate variability, as mentioned earlier, is really is still facing a challenge. We do not have a good modeling approach to characterization of the within substrate variability yet. And from the extrapolation from the adult BE to pediatric BE, generally speaking, we have made some progress as shown by the FDA presentations. However, we do hear that we should be very careful or cautious to extrapolate BE establishing adult to neonates. We need to have to consider the age factor that we can divide the pediatrics patients into appropriate age groups to mitigate the concern. All with this kind of challenges, we do have appreciated a good array of solutions and opportunities for the modeling field to have. Certainly, we can sometimes we can incorporate the gastric emptying and supersaturation precipitation into the equation to evaluate the food effect, and we can make improvement to the in vitro methodologies, especially dissolution method, and to better inform the modeling outcome. It's more like a mutual feedback thing. We can use a good in vitro method to serve as the good input for the PBPK modeling. In the same time, we can use the model to inform the good identification of in vitro dissolution method and also RVE PBPK approach. I heard terminologies like a model-based identification of biopredictive dissolution profiles and critical quality attribute under their corresponding B spaces. We do have heard there is some need for the improvement to PBPK softwares. Sometimes the software may give different outcomes, and there's nuances we need to figure out. And from the IQ consortium, and I learned that we can using the de novo mechanistic absorption models to study the food effect, and they have a stronger impact on the BCS2 and minimal effects on the BCS1 and 3 drugs. For the MR products, modified release product, we can certainly use in vitro and in silico plus in silico approaches for BioWaiver. That could represent a future trend in the modeling field. We have also talked about modeling approaches in terms of bottom-up versus middle-out approaches, and when to have these approaches is kind of a philosophy the community needed to figure out. And certainly also beyond the challenges, opportunities, and we have heard some need for the standard. What would be the best reference profile for virtual BE simulations? What would be the evaluation? What will be the best practices to evaluate the formulation difference? So I heard that leverage any data for other formulations will be very helpful in this regard. So certainly model is the integration of knowledge and experience, should also be both purpose and risk-based. We cannot figure out, we do not need to figure out all of the physiological parameters behind a PBPK model to substantiate it into regulatory use. That's the first part of today's session. On the second part, we touched upon a very intriguing topic first rolled out from this workshop, which is the model master file. Model master file, we do have, since it's the first meeting, we do have a debate on what is model master file, what can be shared, what are the key elements of a model master file. So if there's incremental knowledge or advancement in science in model, how can we improve the model master file? How to share a model master file? Who should be the hosting entity? And what will be the operational aspect of a model master file? Certainly, we do have, out of many different opinions, we do agree that since the model master file is defined for regulatory use, regulatory acceptance is the key benchmark to qualify a model master file. There could be many different types of model master files. There could be lead model master file, parent model master file, and there could be a descendant model master file, upgraded model master file. I think all the things we need to leave for time on the future practice. With that, I want to conclude my summary of today's workshop session. And I'm sure I have left something important out. If that's the case, my apologies. With that, I want to, keep, the agency keep communicating with our industry colleagues, with our colleagues in academia. Together, we form a very harmonious, productive, effective ecosystem to keep expanding the envelope. And finally, my sincere thanks to your contribution and your patience and your time. With that, I want to turn the podium to Dr. Robert Lionberger for closing remarks. Rob, in case you're talking, we cannot hear you yet. Probably you need to unmute yourself. I think there could be some tech issues. Anyway, I really appreciate your patience and time. So let's see if we can get the issue resolved shortly.
Hey, Rob, this is Jim. We can't hear you. We see that you're unmuted, but we just can't hear you.
Rob, do you want to use the phone call? You can switch the audio. But if you click on the event info at the top, you will see the call-in number. If you need a second to call in, that's fine. If you go to the event info at the top of the screen, it has the call-in number. Yeah, I think Rob is calling. Just bear with us for a second. So can we use this time to do a survey for our next workshop in terms of making it physical or virtual? So I see we still have over 100 people there. If you can tap in your preference for FDA CRCG future workshops, your preference virtual versus physical or a combination, that would be great. So you can just tap in the word. I see there already is some responses. Rob, you need to unmute yourself from the computer as well. I think I see you are still on mute. Yes.
All right, can you hear me now?
Yes.
All right, so, sorry for the delay. Just a little late afternoon computer change here. So I want to conclude the meeting by thanking all of our speakers, thanking for you bearing with our audio changes and my technical difficulties. Really appreciate all of our great speakers that we had participate in this workshop. And especially want to thank FDA's organizers, especially Andrew Babiskin, who did a lot of the logistic work. But I also really want to recognize our Center for Complex Generics. And the work that Jim and Anna have done in this is really amazing. This is our second workshop with them. We have another one coming up later this year. But this workshop and the goal of developing and collaborating with industry to implement new technologies is directly in the Center for Complex Generics goal, right? How do you get these advances in modeling and simulation, things we heard of across the industry, into the generic drug pipeline and infrastructure? And I think this is really important and really helpful in the future for the development of complex generics. And, as I think about the life cycle of how we use modeling and simulation, probably the first step is models are used by FDA. Our office, we're big consumers of these because we use these when we're developing our approaches for bioequivalence, our recommendations, evaluating applications. So we are users of these tools, so we have firsthand evaluation of how useful they are. I think the second step is applicants have the opportunity to use these during pharmaceutical development, right? What formulation do I develop? What dissolution profile should I target before they do in vivo studies? And you don't need FDA's permission to do that. And I think it's really valuable to hear from industry that approach works. So the more these tools are used in the space of pharmaceutical development, that's really the step toward moving them into the application, right? If you won't use them, if you're not using them themselves, then it doesn't make sense. I'm going to submit it to FDA before I even use it myself. So we really need evidence that applicants are using these tools in their own development processes. We're using them internally. So the next step after that, and that's where we talk about the infrastructure here and the model master files are, you've reached the point where you want to bring some of this model into the discussion about, in the generic space, am I bioequivalent to the brand product or not? And I think if you do that, that's really where we are now. And we had a great discussion on the model master files, talking about the infrastructure for doing this, talking about the understanding, really identifying the mechanism to do that. So the mechanism is one part. The second part also is the science. What scientific and regulatory questions will the submission of a model really help you address? And I think this is something very thoughtful that you saw examples during the workshop of places where that might be useful. And I think this is something that will evolve. You know, it has to face the test of reality. The models have to solve real regulatory problems that recur in application submission, right? If it's something where FDA uses the model to decide what study to request, right, not everyone doesn't have to repeat that. But if it's something that in your submission or for your product, the model is useful, then there's something that has to go into the specific application. And we want to build out a really good infrastructure for doing that in an efficient way. So really appreciate the forward-looking aspects of this workshop in that. And so with that, again, I'd like to thank everyone who organized, participated, especially all of our distinguished speakers in the fantastic panel discussion. And want to remind people that the recordings for this will be available in 3 weeks. If you register, you will get an email when the recordings are available, so don't bother the Center for Complex Generics about the recordings until then, but we will have full access to all of these. So again, thank you very much and have a good evening.